So you are all very welcome. Just allow me to share my screen, which I should be able to do very smoothly by now, but sometimes. Right. And I will just ask one of my fellow panelists, could you confirm for me that you can see it on presenter view?
Yes, please, we do.
Wonderful. Well, thank you all very much for joining us for this webinar. And I know that because you have joined, you've obviously seen the link somewhere. So you've seen one of the adverts and you've already registered via the link. But perhaps you were wondering, what is it that we're going to do during these talks? What exactly do we mean by a pharmacometric discourse? And why are we launching yet another seminar program when there may already be different seminars in existence in different parts of the world? So before I introduce our first speaker, I thought it would be helpful just to explain very briefly who we are, what we do, why we're having a seminar program, and how you can potentially get involved. So, who are we? We are the Uganda chapter of Pharmacometrics Africa, and many of you will be aware that until really about the last 5 or 10 years, there were quite significant gaps in the training and retention of pharmacokinetic modelers within Africa. And that had a bit of a knock-on effect, because what it meant was that our stakeholders, the decision makers, the clinicians, people didn't really understand the use of modeling. And because of that gap in their knowledge and the lack of local networks, there tended to be a low priority for regional disease threats. And many times data sets were exported out of Africa. And recognizing some of these critical strategic gaps, Colin Pillai and others in 2018 established Pharmacometrics Africa, which has the specific vision to address and respond to these targets. And since that time, more than 500 scientists have been trained across Africa. And if you look at where the hotspots are on this map, you'll see that also there are some other strategic priorities that emerge over time. And really excitingly, in this past year, we've launched the Francophone Africa section of Pharmacometrics Africa. We continue to expand and diversify to make sure that nobody is left behind. But as we all know on this call, Africa is not a country. And so within the different regions, certain regional hubs are emerging, which have really quite specific needs or specific areas of focus. And because of that, in 2021, several of us who are on the call today led the founding of the Uganda chapter of Pharmacometrics Africa. And the QR code here takes you to our webpage. And some of our first activities involved stepping back a little bit from the detail and looking at what people around us needed to know about pharmacometrics. And that led us down quite an exciting journey where we used other techniques. We used creative arts and film and photography to try and demystify some of our techniques. And our films are available on our YouTube channel, which you can link to here. And the aim is that we create a community of practice where people understand what we're doing and why it matters, and why they can trust our results. In terms of the team, I was very excited as I put this slide together, because back in 2018, around about the time I first met Colin Pillai, we didn't really have a pharmacometric group in Uganda. And indeed, some people told me that this was quite aspirational, that it might not actually be possible. And yet, as we look down these faces, several of you are on the call today. I'm sure you are delighted to see yourself looking so vibrant. But we actually have a really strong pipeline of researchers coming through. We're bringing together multiple intersecting grants and programs. We all have slightly different areas of expertise. So in this mix, we have clinicians, physicians, clinical pharmacologists, mathematical modelers, pure mathematicians, and we all kind of overlap in our areas of expertise, which makes the group even more vibrant. We're not just within Uganda, which is very much at the heart of Africa, but we actually have an expanding collaborative network through East Africa, across the continent, and actually worldwide. And our partners include academia, not-for-profit, industry, and regulatory sectors. But what do we do? So in a nutshell, We aim to make sure that every individual has access to the information they need to make an informed choice about their health. So when that means somebody needs a medication, we want to make sure there's the evidence to support how we use that drug in that patient. So we respond to community priorities. We do trials and pharmacokinetic studies, and we emphasize the right sample from the right patient at the right time point. We've spent a lot of time establishing laboratory infrastructure, and we have a lot of support from our collaborators in that. And of course, we have the pharmacokinetic modeling very much, and that's going to be the focus of these seminars, but you have to see how it fits within the context. And as our modeling gets more advanced, we have to make sure we can bring the communities with us and explain what we're doing. So why have a seminar program? Well, actually, whilst there are many seminar programs that do exist, we felt that there was quite a strategic need, because around us, if you think about who you work with, how many times do you have to explain to somebody what you do and why it matters? How many times do we get asked the same questions? If you use PBPK modeling, for example, how can you trust that? And what does it really mean? And aren't you as well just doing the trial? There are many questions that come up again and again. And what we want to do is we want to be able to share really good examples of best practice where we've taken a local priority, we've used modeling techniques to understand that, and then we've moved forward perhaps to either further work or making some kind of recommendation. So in terms of a target audience, yes, those of us that are doing the modeling ourselves, those of us that are interested in potentially getting into modeling, but also please do feel free to invite your clinical colleagues or people in your national drug authorities, people around you that you think might actually benefit from knowing more about what we do. And obviously you know how, because you've already registered, but people can still register via the link for next time round. And we're also recording today's session, and we plan to host these on our website and on the At the Equator YouTube channel, so that we produce an archive of resources. And the whole idea is that we produce something that is of benefit to all of us. So please do come to us with your suggestions of topics. Please engage with the webinar. So how can you be involved? Well, there are many ways. The first thing you've done is you've signed up and you've joined today's seminar, and you are more than welcome. We will be having a seminar on the first Tuesday of every month. And approximately every third or fourth will have a specific PBPK, physiologically based pharmacokinetic modeling flavor. We will be reaching out to certain people to give seminars, but if you are aware of somebody, or if you have in your hand a really good example of where modeling has been used for a locally relevant problem, please do come to us, because this might be an opportunity for you also to share some of your findings. We hope that it provides a little bit of a forum for input and mentorship. So if you're on the call and somebody asks a particular question and you've got some response to that, please join in. Those of you who are more senior, as you get to know our group and you interact with us, perhaps you'll see other ways you can get involved. For everybody, as I say, this is not a display. This is not us giving very polished, perfect seminars, but it's providing the opportunity to have that conversation, that discourse, as we put in the title of the program. And your responsibility is to ask the questions you have. I tell everybody that I work with that there is no such thing as a stupid question. If you're on this call and you have a question about any aspect that has been discussed, please put it in the Q&A. There is a chat button and a Q&A button. If you can, remember to use the Q&A, because it's easier to give you an individual response to that. Sometimes in the chat, things get a little bit lost. You can also, beyond this, as I said, the recordings will be available and open access, and you can continue to follow and engage with our social media. So that is a very brief overview of what we are doing and why. We are delighted that it is the first session, and I have got great delight in inviting our postdoctoral modeler, Ida Kawuma, who's going to explain an example of just why we need to use these techniques. So Ida, when you're ready, over to you.
Thank you, Katrina. Please confirm that you can see my screen and hear me.
I can see your screen and hear you, but you are not yet in presenter mode.
Great. And now?
You are. Now you are perfect.
Okay. Let me just get, okay. So thank you, Katrina, for that introduction. I'm delighted today to take you through some of our work looking at the pharmacokinetics of drugs used to treat drug-sensitive tuberculosis in breastfeeding mother-infant pairs. So this is an interim analysis of rifampicin, and I'm going to walk you through the work that we've done and show you how we applied pharmacometric techniques to get to the results that we have today. Okay, so I'll begin with a little background. So approximately 50% of women worldwide will need to take some kind of medication while they're breastfeeding. And for ladies in low and middle income countries, breastfeeding really is the only acceptable, feasible, affordable, and sustainable and safe option for infant nutrition. And most of the cases that we have of tuberculosis, unfortunately, occur in these scenarios. And so we have a large quantity of women who are breastfeeding, but who are needing to breastfeed, but who are also on anti-TB medication. And largely no data exists to describe the transfer of drugs from the breastfeeding mother to the breastfed infant. Hold on. And so the rationale for wanting to describe drug transfer in breast milk is at least more than twofold. So first of all, it's important because when infants are exposed to certain drugs from the breast milk, in this case, the TB drugs, it's possible that they'll get adverse drug reactions. These reactions might be immediate, they might be acute, or they could even be prolonged. Also, if the infant is exposed continuously to low amounts of anti-tuberculosis drugs in the milk, through the milk, these drug concentrations might select for drug resistance. And so if they end up needing this medication, they might be resistant to it. But also, it is possible that the concentrations that the infants receive through breast milk might offer some kind of protection, some sort of prophylaxis. And so that is why it is important for us to describe the transfer of these drugs from the breastfeeding mother to the infant. So we put together a milk study, which stands for maternal, infant, and lactation pharmacokinetic. And in this study, our aim was to look at the pharmacokinetics of first-line TB drugs in breastfeeding mother-infant pairs. And we also wanted to define that transfer to the infant through breast milk. So some of our primary endpoints were wanted to look at the maternal plasma and breast milk concentrations at different time points, at 0, that's pre-dose, at 2, 4, 6, 8, and 24 hours post-dose. We also wanted to look at what the infant blood concentrations are, especially at maternal trough, that's at pre-dose, and also about 8 hours post-dose, up to 8 hours post-dose. We also wanted to calculate the area under concentration time curve, which gives us the overall exposure within certain 2 points, and also therefore be able to calculate a breast milk to maternal plasma ratio. So it's simply milk to plasma ratio is what we wanted to be able to define. So for the milk studies, what was our criteria? We included, obviously, willing mothers, those who are were willing and also would let their children, their infants be part of the study. If they could give consent, they had to be over the age of 18, and they had to be breastfeeding or pregnant at the time of enrollment. If they were pregnant, we then waited until they had delivered, had begun breastfeeding, and then took off the study samples. Of course, if a mother was severely ill or an infant was severely ill, we excluded them because it would be difficult for them to take part in study activities. I would like to draw your attention to sample size calculation. So oftentimes this is a very sticky point for studies. And since they're pharmacokinetic studies, I'm sure many of you have questions about, is the sample size enough? What sample size should I use? And like I said earlier on, there's hardly any data on transfer of drugs, of TB medication to the breast milk. And this was the case when we were designing this study. There was no prior data to inform sample size calculation, hardly any. In fact, for rifampicin particularly, the recommendations, the guidance we use in breastfeeding mothers comes from a single publication in 1969. Now that's 50 years ago. That's when Neil Armstrong walked on the moon, Apollo 11 landed on the moon. But we are using recommendations now that were 50 years ago. Moreover, if you go to that snippet on LactMed to look for that information, you'll realize that in that publication, they didn't give details about the study, they didn't give details about the patients. So you can see that 50 years on, we have recommendations which have come from a single publication, and we can't be sure about how well this study was conducted. So you can see that there was really need for us to do these sort of studies. I mean, you can imagine if you were using technology from 50 years ago, it's been phased out. But unfortunately, we are still using such recommendations for medications in breastfeeding mothers now. So what did we do? Like I've said, there was no data. So we conducted what we call a stochastic simulation and estimation exercise. So what we did was get a previously published model, and then we made some assumptions. We said, if drug was to go to the breast milk, was to transfer from plasma to breast milk, what do we do? How do you describe that? So we added what we call an effect compartment and two new parameters, and I'm going to describe those. We added an accumulation factor and an equilibration rate constant. So let me bring out my pointer and then hopefully go. So this is just an illustration of the sort of model we got previously, which was published. So meaning the drugs are taken orally, and then there's a bit of a lag, or we can call it a transit into the gut. When you swallow the medication, then it is absorbed into plasma and it's cleared. And we take samples from this, what we call the central compartment. So that's the kind of model we got. And then we made an assumption. And so we extended that model and said, what if drug came from the plasma and went into the breast milk? So we added two new parameters, which we call an accumulation factor, which simply shows it's basically the milk to plasma ratio, how much of the drug accumulates in the milk vis-a-vis in the plasma. And then an equilibration rate constant. How long does it take? So we added two new parameters, and this is the sort of the illustration. So this is not real data, but this is just to illustrate to you what would happen. So if a drug has a high breast milk to plasma ratio, that means it accumulates. There's high accumulation in the breast milk. If the accumulation factor is low, that means it does not accumulate as much in the breast milk. So that is one parameter that we added. Another parameter that we added was the equilibration rate constant, which is simply the delay. So there's how much it accumulates, but also how long does it take? Does it accumulate instantaneous? Does it get there instantaneously, or is there a delay? So these two parameters are what we added onto the already published model. And then we ran simulations using that new extended model. And so we ran 200 simulations, and in those simulations, we're simply exploring how many individuals do we need to precisely estimate our parameters. Do we need 15? Do we need 20? Do we need 24? And that was looking at a particular sampling schedule. If we sample at these different time points with this number of participants, are we able to precisely estimate the parameters? So running that simulation exercise led us to the conclusion that we needed at least 15 mother-infant pairs over around an 8-hour sampling period to be able to precisely estimate our parameters. And then we kept the infants to we recruited infants only less than 12 months. And that is because at about 1 year, most infants have been weaned and their sustenance is mainly they're eating food, and breast milk is just the supplement. And so as opposed to much younger children. So at this point, I just want to highlight the kind of treatment we're talking about. The first-line treatment for drug-sensitive tuberculosis comprises a 6-month treatment period where we have 2 months intensive phase where we use 4 drugs, rifampicin, isoniazid, ethambutol, pyrazinamide, and then a follow-on period where we use 4 months and you're taking 2 drugs only. That's rifampicin and isoniazid. And so I'd like to then take you through what our study design was like. So for our study design, like I've said, we enrolled pregnant or breastfeeding women who are requiring treatment for TB. And so for the mother, we took a matched plasma and breast milk sample at 0, 2, 4, 6, and 8 hours post-dose. Those were sort of required samples. But we did ask the mothers if they stayed nearby. If it wasn't too difficult, we told them to come back the next day, and all of them, majority of them, in fact, did come back. And then we took a sample at 24 hours post-dose. So for the infant, we took a heel prick dried blood spot at maternal at time 0 for when we're taking the time 0 sample for the mother. And any time between this 8-hour period, we took another infant sample. Okay, so that was visit 1. And ideally on visit 1, that was during the first 2 months of TB treatment. So participants were on all 4 drugs. Then we asked them to come back for a second visit during the continuation phase of their treatment when they were on only 2 drugs, and we did the same thing. We took a breast milk, we took a plasma and breast milk sample at 0, 2, 4, 6, 8, and 24 hours post-dose. But you'll note that for some of the women at the time of recruitment, at the time we enrolled them, they had already gotten off the first phase of the intensive phase of TB treatment and were already on the continuation phase. And so I just wanted to highlight that for some women, we only got 1 visit if we got to them, if we recruited them when they had finished with their intensive phase. So that is that was the study design. That's how we got to the numbers. And right now, I just want to walk you through rifampicin in particular. So in our protocol during the study design, we put a clause for an interim analysis. And the reason being was we wanted to check if the assumptions we had made during the trial design were sound, and whether the design we were using was still going to be able to answer the questions we wanted. So the interim analysis, why we did it, it was aimed to validate the proposed milk antituberculosis study design, the feasibility of the sampling schedules we had chosen, the feasibility of the recruitment and the mothers and their infants, and also to validate the proposed sampling times. So that's why we took this interim analysis. So after we've enrolled the first 6 mother-infant pairs, once they concluded their visits, we then took on this analysis. And so blood and breast milk concentrations were quantified using LCMSMS at the University of Cape Town, Division of Clinical Pharmacology. And then this analysis was done using is a non-compartment analysis, and we used phenixquinone to do it. So I'd like to just highlight the 6 mother-infant pair, the 6 mothers that we see. These are concentration time profiles. The dotted lines are the breast milk, and the solid lines, or that you may call them dashed. The dashed lines are breast milk. The solid lines are plasma concentrations, and the orange vis-a-vis the green are simply on the 2 different visits. So as you see, for mother number 2, we only got samples from 1 visit, whereas the rest of the mothers, we had samples on 2 different visits. So we had 65 paired samples, breast milk and plasma. 18 plasma samples were below limit of quantification, and 27 breast milk samples were below limit of quantification. The purple and black lines, as you see, are the limits of quantification for plasma and breast milk. And you can see these samples all being below the limit. Then for the infants, remember we said we took a heel prick blood sample. All their concentrations for the 22 of them were below the limit of quantification, except 1. And that 1 sample belonged, the infant who had a measurable plasma sample belonged to the mother number 6. Okay. So what are the findings of our non-compartment analysis? What I want to show you here is that we simply did it in plasma and in breast milk, but also we did it on the two visits, the two separate visits. So what I want to highlight here on both occasions, first visit, second visit, is that we see that the time to achieve the maximum concentration in the plasma, in the breast milk is higher than that time it takes to achieve than the time to peak in plasma. So remember earlier on when I said that we added two parameters, there was an accumulation factor and a delay factor. This sort of confirms that the peak in the breast milk is realized slightly later than the peak in the plasma. So the concentration of drug, it first peaks in the plasma, and after some time, it sort of like takes a while to transfer in the breast milk to the breast milk. The time to Tmax then is later. So that is clear in both visits. Obviously, the maximum concentration is highest in the plasma, like you saw from these graphs, and also the area under the concentration time curve is obviously higher in the plasma. So the results, so from that, after we had the area under the concentration time curve, we then calculated a milk to plasma ratio, and that was simply the area, the AUC 0 to 24 in milk over the AUC in plasma. And we saw we got a ratio of 0.16 visit 1 vis-a-vis a ratio of 0.18 in visit 2. So all in the same direction. You have breast milk, but lower concentrations. You have the drug, but in lower concentrations in the breast milk. And we also see that there's delayed plasma to breast milk distribution of rifampicin. So again, that shift to the right slightly. So in conclusion, I would like to say that modeling techniques can help inform study design aspects, especially in scenarios where there's very prior data. So there was not much to stand on in the way of sample size estimation. And so we used a modeling technique. We got a model that was already available, made some assumptions based on physiology and understanding, and then extended that model to sort of include a breast milk compartment, an effect compartment. And so we see that modeling here was really a helpful technique. And then within the study design, we included an interim analysis, and this interim analysis gave us the opportunity to verify whether our initial study design's assumptions held or not. And in fact, if we found that the assumptions didn't hold, it would have been possible to then modify our protocol design. But based on this analysis, we did not modify our study protocol. So what are next steps? Like, we have concluded enrollment and recruitment of the 20 mother-infant pairs. So we are now going to develop a full population PK model using data from the mother. So in the beginning, like I said, we simply extended a model. It was an assumption, but now we're actually going to develop that model with plasma and breast milk concentrations. And with that, I'd like to acknowledge the study participants, the mothers. You'll realize that these were breastfeeding mothers. Some of them, the infants were very, very young, but they came back even 24 hours after. So the back and forth, they allowed us to prick their infants even when they cried. And also, I'd like to thank the milk study team, the UCT Division of Clinical Pharmacology, and of course, our umbrella body, Pharmacometrics Africa, and Katrina Waite, who was the PI, and my co-modeler, Francis Ojara. And with that, I'd like to thank you for your attention and open up for questions.
Wonderful. Thank you, Ida. Of course, I've heard this presentation before and have been deeply invested in all the stages of the work, and yet still, I think it's an extremely good and clear example of just why we need to use modeling both in the study design stage and the analysis. And I think it shows that modeling isn't an afterthought that is done at the end of a study. So I'm going to ask you a little bit about that. I'm noticing that there's a couple of questions are coming up in the chat. and while some other people do type their questions, I would just ask you, Ida, how would you compare this experience of having been, I'll try not to ask it in too loaded a manner, how would you compare the experience of being involved from design stage and at the interim analysis, and now you're going to be building a full model? How does that compare to when somebody completes a study and they give you a ready-made complete data set? What are the compare and contrast between those two scenarios?
I think one of the key advantages, if I may just go back to these slides, I'll go back to the study design so I can explain better here. Okay. There, I think my experience being involved later as a modeler is oftentimes clinicians might design a study and for some reason decide on the sampling time point. So let me say someone might decide on 0, 4, 12, and 24 hours. And I mean, sometimes the decisions are based on the funding that's available. Sometimes the decisions are based on the perceived, will you be able to sample that much? Will you be able to draw that many samples? You know, so there are different decisions that, reasons that might drive the sampling schedule. But oftentimes when someone comes to you with a completed study and they bring you a data set and say, analyze, sometimes you almost want to say, you could have sampled better, you know, you could have made more meaning, you could have distributed your sampling time points in a more meaningful way, you know, or you would have taken, for example, sometimes people, clinicians come and they've concluded a study and they've taken sparse samples, meaning only either 1 or 2 samples from 100 participants, you know. And so what then happens is, if I can show something, if someone has taken sparse samples, for example, someone might bring a data set and they have a cloud of samples very close to the 24-hour time point, you know, and then they have maybe a cloud of samples very close to the beginning of the study. And in between, there's nothing. So they've sampled 100 patients, but you can't make a lot of data, a lot of meaning from that sparse sampling. Whereas if you had been involved from the beginning, you could have said, look, instead of sampling 100 patients, maybe let's sample 20 and sample them more intensely so that you have a richer profile. So I guess what I'm saying.
There is actually a question asking us how we decided on the sample size and the sampling time points relative to dose. So I think you're already answering that, but specifically to this study, how did you decide to do that sort of up to 8 hours and 24-hour thing?
Yeah. So to answer your question, one is the drug. There had been some literature about rifampicin and sort of some information about when it peaks, the time, the Tmax. So you want to sample around there, you want to sample before, and also you want to be able to describe the slope of the drug. So based on, I mean, based on the half-life of the drug, that can inform your sampling. But also the advantage of the modeling technique, stochastic simulation and estimation, is that when you're running these simulation exercises, when you're testing if 20 people are adequate or whether 32 people, I mean, when you're looking for the number of people who will help you adequately define your parameters, you can also simulate the sampling schedule. You can play around. So you play around and say, if I sample at 0, 2, 4, 6, 8, vis-a-vis 1, 5, 10, 13, 16, how best do I estimate, how precisely can I estimate the clearance, for example? Because if you want to estimate the slope, you're going to need some time points, you know, in that phase. If you want to estimate the absorption parameters, you need some time points within the absorption phase. So what I'm going to say, it's to answer your question, there are many things you take into consideration: the half-life of the drug, the feasibility of the sampling itself. For example, if you have exceedingly sick patients, let me say they're in the ICU, it will be hard to say, you might not be able to sample so frequently. But to answer your question, you can play around the sampling schedule. with the simulations. So that is really, really helpful. But also you have to think about a few other parameters of the drug. Yeah.
Thank you.
So Katrina, I think just to end, I just want to say that involving a modeler early will improve. I think what I can say, it will improve the quality of your data and what you can infer from that data.
Thank you. Because obviously sometimes it is a bit of a loaded question, but I think there's nothing worse than when you get given a data set and there's just some flaw and you just think, oh, it almost feels like a missed opportunity. And at times it can almost feel unethical that somebody has trusted you with their samples and their data, and yet your results are not very conclusive because the study design wasn't quite there. So I think it's just a very important reminder to integrate the modelling into what we're doing. I see several questions in the chat, and we're responding to some of them as we go, and I'll also verbalize some, but I also see, Jessica, you have your hand up. So I'm going to unmute you and allow you to speak, I think. You should be unmuted. Oh, you're muted again.
Hi, sorry, thank you. I didn't actually realize my hand was up, but I had posted a question in the chat. And I was just wondering in this sort of setting with this type of drug, how auto-induction by rifampicin might have influenced the first visit's results, or did you take that into account and only do it later on in the first 2 months of treatment? Thank you.
Thank you, Jessica, for such an interesting question. So one of the things we ensured to do is that even when a mother started TB treatment, we only sampled at least, you had to get to 2 weeks. And by that time, the drug should be at steady state, and also rifampicin autoinduction ideally should have gotten to about maximum. So we tried to, we enrolled you, and if you had just started your treatment, we did not sample until you were at least 2 weeks after your medication. Yeah. But to answer, another something else that's an advantage of modeling is that we can indeed incorporate this during our next steps when we are developing a PK model. So if we cannot, if we feel that we can't describe the kinetics without the aspect of auto induction, we can then account for that and write that into the model.
Fantastic. And I think that leads on to another question about some of the complexity, because a couple of the delegates have almost pointed out that nobody's going to just be on rifampicin. You know, they will be on it as a component of anti-TB treatment, but also at least 60% of them will have intercurrent HIV in addition to any other medical conditions. So there's 2 linked questions here from Brenda, who asks whether when we're in modelling, do we factor in other medications and also things like general health and BMI? And Sagar Kore asks if there are any information about drug-drug interactions and would that affect the PK in either the mother or the infant? So I think those 2 can probably be handled together.
Thanks for your questions. Again, like I've said, and I'll begin to sound like a broken record, but the advantage of modeling is that indeed, when we are developing these models, for example, we are defining we have different compartments, you know, and one of the key advantages of modeling is if there is a patient, a variable, we call them covariates. If there is a variable that we think affects clearance, affects absorption, you can include these onto the model. So one we always include, for example, is the weight. Your weight will most likely, your clearance can be a factor of your weight. Your volume of distribution can be a factor of your weight. So again, like I've said, for the non-compartment analysis, we did this because it was quick and suitable to answer the question at the time. So the NCA, the non-compartment analysis, was sufficient for the interim analysis to see if we can then continue with our study. But now that we've gotten all our 20 mother-infant pairs, we have more information. We have a wider range of demographics. So now we can begin to include things like weight, body mass index. We can include fat-free mass. So we can test all these as a covariate affecting aspects like clearance, affecting aspects like the volume. You can add, for those of you who have talked about drug interaction, you can look at visit 1 vis-a-vis visit 2, because visit 1, you had the formulation with 4 drugs combined. Visit 2, you have a formulation with only 2 drugs combined. So already you can do visit 1 vis-a-vis visit 2, knowing that the difference there, so you can account for formulation. In terms of drug-drug interactions, we also did collect information on whether a mother is living with HIV, because we do know that HIV, TB, HIV confirmations are very high. So we also collected information on whether a mother was living with HIV. And if she was, again, if we look, if you see if you see a very big, what I'm trying to say is that we can incorporate these drugs as a potential covariate onto the model and test for their effect. So covariates like body mass, age, drug interaction, we can all incorporate that into the model. And that is one of the advantages of modeling, which you cannot do with an NCA. You can't do that with an uncompartmental analysis. You can, but you'd need loads of data of people in both arms. Yeah.
And I would say actually that's one of the strengths of modeling, because if you think of all the diversity in our participants, we do know that they're going to be on other drugs, but the exact combination within that individual, they might be quite unique. And yet modeling allows you to do almost like virtual clinical trials. You can adjust various different things, you can simulate, and that's actually really good. The more difficult the population gets, the more unique each individual gets. Modeling has a particularly special role, I think. And I think hopefully we'll see that in some of our future seminars. I'm also seeing another theme of questions relating to the infant. And again, I can see Frances working very hard behind the scenes typing questions. So one of the questions was to do with the assay itself and how well validated and developed that is. And actually, I don't think the samples will have been BLQ, below limit of quantitation, because of the assay, because all of the assays are validated according to there's quite a rigorous way of validating assays. So if you look at the FDA guidance for bioanalysis, there's something like 9 steps that you have to progress through. And when you already have an assay for one matrix, so the matrix might be plasma or something, if you want to bring in a new matrix, for example, a dried blood spot or a breast milk sample, you do what you call a partial validation, but you won't actually proceed until you've done that. And in every run of analytes we do, you have high, medium, and low quality control as well as standard curve. So you know that the assay has passed. So I don't think it's methodological, but there were some quite interesting questions. One individual has asked about if there were any sort of challenges in sampling from the infants. And Amaka has asked a very good question, actually, that you will have seen before. Because all of the samples were BLQ in the infant, except for that one where we think the mother had taken an extra dose, does that mean that we subsequently have not measured rifampicin in those infants? Does that mean that we stopped sampling from the infants? Does that mean we've stopped measuring rifampicin in those infants? So logistics, and did we change our study design on the result of it?
Sorry, I was trying to unmute myself. Yeah, so thank you for a very interesting question. I would like to draw your attention to the fact that we said very early on, we are looking at all the first-line anti-TB drugs, so rifampicin, isoniazid, ethambutol, pyrazinamide. So whereas for rifampicin, it was mostly BLQ in the infant, that does not negate the fact that other drug might be in quantifiable amounts. So to answer your question, no, we did not stop sampling in the infant, because like I've said, we hope to do this for all drugs. So whereas rifampicin might be BLQ, you might find that when we run the assays for the other drugs, there's quantifiable amounts of drug there. And then also, there's a lot of variability for rifampicin. And the fact that we didn't get it in the first 6 mother-infant pairs does not negate the fact that it might actually be there in other infants. So I think that to sum it up, we did not stop sampling, but we did revise our guidance to the mothers, because we suspect, and let me just go there. We suspect that bear with me. So you see, this mom had quite high concentrations, and it's the fact that her pre-dose concentration is very high. In fact, it's higher than many others. We suspect this mother inadvertently took a dose of her medication before she came to the clinic, because sampling on the day of sampling, all medication was directly observed. So we suspect that this mom took her pill back at home, came to the clinic and said, when we asked her, did you take your medication? She must have said no. But in fact, that concentrations at time 0 give away the fact that they were very high concentrations. So we suspect she probably took took another pill very soon after at the clinic, and that's why we see them very high. So one thing we did go back with our study nurses was to say, look, please inform these mothers not to take their pills, their medication on the PK day. It will be directly observed, taken in the clinic. Yeah.
Thank you. And of course, we haven't actually assayed all of the remaining samples. We've completed the clinical follow-up, and as we look at the data that are coming through, we can decide if we do actually go ahead to measure every single thing in every single sample, or whether we're somehow more selective. So we haven't quite finished all of that yet, and it's a good reason for doing the interim. A couple more questions, Ida. Henry and Zama has asked about any pharmacodynamic endpoints and how we might address those. And Ronald Kadada I think it's an overlapping question, but what are the main factors that might affect the transfer from plasma to milk, and which ones have we tested during this analysis? So I think some of it's pharmacokinetics, but some of it's this sort of PK/PD relationship and individual variability, and how far can we go with the kind of work we're doing?
Yeah, thanks, Henry. So because this was an observational study, so we didn't intervene in any way. We're not linking the concentrations to any PD outcome, we didn't take we haven't rigorously collected information on things like safety, adverse events. Of course, if someone came with an adverse event, were obliged to care for them, but this study was mostly we just wanted to focus on the pharmacokinetics. And since it was observational, these were patients who we did not give them the drug, they were on their medication. So we just wanted to observe, is what I'm trying to say. But in the event that we collected PD, some sort of PD marker, having a pop PK model, you can in fact describe a PD model. You can have pop PK model with the breast milk compartment included, and then you can even further extend that to a PD model if we had taken those kinds of markers. Then to answer Ronald's question, so you have to think about, of course, factors that might affect the transfer to breast milk is the lipophilicity of the drug, because we know, for example, that breast milk is so if a drug was largely lipophilic, you can already assume that ideally it should transfer more than a less lipophilic drug. We are also considering, it's not well described, but we're also wanting to investigate, not in this study, but it's also a possibility, whether a mother whether it's fore milk or hind milk, in that the first milk before a mother breastfeeds, and then when she continues breastfeeding, the composition of that milk is also different. That is not something we have explored here, but it's something also that we think can explain how much breast milk, how much drug transfers. We think also feeding, obviously, in that what you feed on, is the mother taking a galactagogue or not, might affect the composition of your breast milk, and therefore it might affect whether a drug transfers more readily or not. The pH of the drug, not the pH, the I forget what it's called, whether at a certain pH, whether it is acidic.
Ionization as well. And,
Ionization, yes. Thank you, Katrina. That's what I was talking about. So there are lots of possible of things that can possibly affect whether drug transfers to the breast milk or not. And like Katrina said in the beginning, these are some of the things that our group is really passionate about, and we think that the more we do more of this work, we're just going to build on that, and hopefully with also PBPK models, our team, like Katrina said, there are PBPK modelers who might be able to tease out these aspects even better than a POP-PK model will do, because with PBPK, you can really input all those constants and define it better.
Thank you very much. There is one question that I'm not going to ask Ida, because I don't think she could do justice to it in about the remaining 3 minutes. But somebody has asked about the mathematical techniques to evaluate the precision of parameters based on the number of patients to conclude the number of patients in your design. And somebody else has asked about, what about sparse sampling if you only have 1 or 2? Can you build a population PK model with those? So what I would say to these 2 attendees, or maybe it's the same individual, this is something that will come up. It will definitely come up, perhaps through a future scenario. I think to do that justice, we would probably need almost an hour. But what we could try to do is find a case study that actually perhaps showcases some of this. And I can see 1 or 2 other comments, but I believe just about everything has been answered live. I want to particularly thank you, Ida, for giving this presentation and kicking us off. I want to thank Gabe and the team at Pharmacometrics Africa, who've helped us set up the platform and have helped us with some of the behind the scenes tech. I want to thank Francis particularly, and Joshua. I've seen you both typing away in the chat, but particularly I'd like to thank all the participants. It's a bit like when you have a party and you're really looking forward to it, but your biggest fear is what happens if we get to the time and nobody comes because maybe people had other things to do. And I think the fact that towards the end of the working day here in East Africa, we have 50 people online, and you've stayed online and you've engaged. This is what we want to see more of. So as I conclude, we will be advertising the next speaker and the next. At some point, we might have like a program, very organized in advance, but we will communicate on a case-by-case example. You will be logging in and registering via the link or the QR code. The reason for that, I think most of you probably are aware of that, it's just an additional layer of safety to keep this a really nice session where we can all engage and we're not at risk of being spammed. So again, any further questions, you know where to find us by email. You can follow the At the Equator Twitter accounts, YouTube channels. Everything that we discuss here, we're recording it. We're not discussing sort of private, hidden, confidential matters. So if you do wish to engage with anything that we've covered today on social media, or come back to us with it, or discuss it with any of your colleagues, or share the recordings with them, please do, because the idea is this is a resource that you can all build upon. So with that, 1 minute to go. Thank you very much for your attention. Have a good day.
Thank you. Bye-bye.